Hello and welcome to the QIMR Berghofer Medical Research Institute High School Lecture Series 2020. My name is Professor Fabienne Mackay. I am the Director and CEO here at QIMR Berghofer. It is my great pleasure to introduce this lecture series on psychology for the first time. We have started this new topic in our lecture series to align with the new senior subject of psychology. Unfortunately, because of COVID-19, we can't welcome you into the Institute as we had hoped to, but we are pleased to deliver to you the series online. Up to 20% Australians, 1 in 5 people, are estimated to have either severe, moderate, or mild mental health disorder. We know that for many people, the risk of mental health problems are higher at the moment because of the changes and challenges brought about by COVID-19. For decades, our researchers have been working to untangle and understand the complex factors that influence our mental health, and in particular, how much of a role our genes and our environment play. In this series, some of our top mental health researchers will examine the thought-provoking question: are psychological traits contagious? They'll explain the role of stress and explore the effects of the current COVID-19 pandemic on our mental health. I hope we will be able to welcome you back to the Institute next year. But for now, stay safe, and it's my pleasure to introduce our first speaker.
Hi, I'm James Scott. I'm the head of mental health program, and I'm a practicing child and adolescent psychiatrist. I'm going to share with you today some thoughts about anxiety. So, in the last few months, everyone has been anxious at some stage. And this anxiety in this time has particularly come about from this pandemic. Now, in Wuhan, in China, is a city of 11 million people. It's crowded. It's seriously crowded. And in December 2019, a strange thing happened. Doctors started noticing that people were coming to the emergency departments with severe viral pneumonia, and it was killing them. The patients coming through became more and more, and the doctors in China started to say, there's a new disease that's coming on. People started to panic, understandably, as we saw doctors and nurses and people in the public of China dying. The Chinese government, on the 23rd of January, instituted a lockdown. This incredibly busy city became like a ghost town. There was no one on the streets. Everyone shut up in their houses, trying to avoid this illness. And we watched from afar, we watched from Australia looking at this, and none of us had any idea what was going to happen. A virus was identified, the novel coronavirus, which gave rise to this illness, COVID-19. This tiny virus we couldn't see was taking lives. 1 in 500 children and adolescents who got this illness died. 1 in 7 older people who got this illness died. This was a terrifying disease. As we watched from Australia, and we saw it spread to Italy, the UK and Korea, and all of a sudden it started coming to Australia. Every 3 days the number of cases in Australia doubled. People were getting sick, and it became a real threat to the Australian population. The Australian government said, guys, we've got to do social distancing, keep apart. But instead of doing that, people went to the beach, just like Australians do. This is a photo of Bondi Beach in late March, and this is what got the Australian government panicking. They said, all right, enough's enough, we've got to stop this virus spreading. The next day, it became illegal to go out in public on the beach. This was Bondi Beach next day, and the lockdown had started, the close down had started. And so all of us have lived with these restrictions that have occurred in the Australian community to stop us getting sick from this virus. And wonderfully, it's worked. We went from the number of cases doubling every 3 days to this flattening of a curve. Cases didn't continue to go up, but it went down. And at this point in time, in May 2020, we're all feeling pretty safe. The anxiety's gone, we're okay. But the most common emotion that people did have during the COVID crisis was anxiety. Anxiety is an emotion we all experience. It's a negative emotion. It's a feeling of worry, of sadness, of irritability, sometimes anger or fear. It's accompanied by physical symptoms like upset stomach, headaches, neck aches, feeling like going to be sick. It's got all these physical symptoms that come on from a arousal of your autonomic nervous system, that fight or flight response. And we get these thoughts of apprehension. Something bad is going to happen. Everyone's experienced anxiety because anxiety is a normal emotion. The question is anxiety, when is it normal and when is it problematic? Because everyone felt anxious as they saw the threat of this COVID virus coming. That was normal anxiety. Anxiety is problematic when the level of anxiety is excessive, when people are so panicked that they can't think, can't function. Anxiety is a problem when it comes on in situations where you don't have to be worried. Sometimes I see kids who can't go outside because they're worried that they might get bitten by a dog, or they're worried about that they might get robbed or something like this. So when there's anxiety at a time when there's no real threat, that's when anxiety is a problem. And finally, anxiety is a problem when it causes impairment. People might feel anxious about going to do an exam, but when your anxiety is so severe that you can't actually sit down and think and do the exam, that's when there's a problem. So anxiety can be normal, but in a number of people, it can get really problematic. Cause of problematic anxiety includes genetic causes, so people have genes that make them more likely to get anxiety. Psychological causes, so sometimes people just think in a way where they tend to sort of worry too much about things. They just sort of have developed this way of thinking, and often that comes from learnt behaviours because perhaps there's anxious people around them or anxious parents. And there's social factors. If you live in an environment where everyone else is anxious and there's a lot of stress going on, like sometimes people living, say, in refugee camps where they really are threatened, there's a lot of anxiety there. So there's a multitude of factors that cause problematic anxiety. The really good thing is we can manage anxiety. When people have problematic anxiety, we can treat this. We can give people information about what anxiety is, and so they can recognise, oh, this pain in my chest, this terrible feeling in my stomach, that's just anxiety. I just need to take steps to manage it. It doesn't sort of lead to them panicking about these physical symptoms they get. Then there's behavioural strategies like improving sleep, improving diet, regular exercise and relaxation strategies are all very effective for reducing anxiety. And finally, there's cognitive therapy. Cognitions are thoughts that people have. So perhaps people get these really unhelpful thoughts like, oh, if I go outside I might get bitten by a dog. And then we teach people to really calculate, well, what's the actual risk of getting bitten by a dog? Oh, the risk's very small. It's safe to go outside, so they can then go and do it. So you package all these things together in a therapy called cognitive behavioural therapy, which is really effective for treating anxiety. So that's how we manage problematic anxiety. So when you think about what's happened with the COVID-19 and the response of the Australian population to this threat, in actual fact, although people felt anxious, only about 12% of people had problematic anxiety, and that's up from a survey that was done 2 years ago where 8% of people had problematic anxiety. So there was a small increase in problematic anxiety in the Australian population, but most people were just fine. They saw the threat, they took steps to manage it, they had confidence the government would look after them, and they got on with life. And that's how most of us are. We manage our anxiety. So in conclusion, anxiety is a normal and healthy emotion. It keeps us safe. We all experience anxiety, and anxiety is there to help us, protect us from doing things that might be dangerous. Anxiety is a problem when it's excessive. It occurs when we don't need to experience it, or it stops us doing the things we need to do, like going to school or going out with friends or these sort of things. That's when anxiety is a problem. Problematic anxiety is really easy to treat. It just needs some strategies, what we call cognitive behavioural therapy, that allows us to manage our anxiety. And so the anxiety, it's common, but it can be managed, and recognising it, getting treatment for it is the key. Thanks very much for listening.
Hi, I'm Lucia. I'm a psychologist and I work as a scientist at QIMR Berghofer. And today I'm going to tell you about something that we study here and I really like. It's mental health and the role of genetics and stress. It is a very important topic. Around half of the people living in Australia, at least once in their life, will have some kind of psychological disorder. It may be depression, anxiety, problems with alcohol use. And today I'm going to focus on depression because if we take into account like not just Australia but people all around the world, around 20%, 16 to 20% people will at least experience once in their life an episode of depression. But when I talk about this, before going further, I would like you to think about what we call the bell curve. Pretty much any human trait that we can observe and pretty much everything that we can study in psychology, including depression, follow this kind of distribution. What does this mean? If we take, let's say, height, like in this picture, and we have a group of people, in this case we have women dressing in white and men dressing in black, and we group them, we put together everybody of the same height, we will see that most people will concentrate, will be aggregated like in the middle. They will be around the average height. Then we will have a smaller group on the left-hand side. They will be shorter people, and then we will have another smaller group on the right-hand side, and there will be the taller people if we compare with the average. And this is something that we see in height, but also in many other traits like IQ or personality traits and depressive symptoms. And this is something good. It is adaptive for us as a species that we can see all this diversity in these traits. As I said, it is also the case for depressive symptoms. When I say depressive symptoms, I'm talking about feeling sad, feeling low, lacking energy, or having problems even sleeping or with appetites, having low self-esteem. And if we ask a group of people in a classroom, let's say, most people will be around the average of depressive symptoms, with, to some extent, some of them. And that's also fine. These symptoms and these feelings are also adaptive to some extent. We will have a small group of people on the left-hand side with very low levels of these symptoms, and then we will have another group on the right, which are people who are experiencing this but in an intensive way. They may feel stuck there, they may feel it's impacting their day-to-day life. And it is in this case when we can talk about depression. So in the end, when we hear a depressive episode, depression and so on, what we are doing is to take into account all this continuum of symptoms and then drawing an artificial line there to just distinguish who may have or not what we call the disorder of depression. And interestingly, if we ask this to the same people 1 year later, let's say, maybe people who was around the average have moved around. Maybe they are crossing this line towards depression, and people who were there are going back to the average. So it's very interesting, and it's very important to understand why does it happen, why people just move around this spectrum around life. So it is very important and very interesting as a scientist to get to understand why we navigate this spectrum, this continuum, why sometimes we may be towards the middle, or sometimes we may be towards the right-hand side and feel depressed. And there is a model that explains this. We call it the jar model, and it says that, well, we are a jar, and then we may be filling this jar with things that are increasing our likelihood to feel all these symptoms of depression and maybe depression at some point. These factors, these things that we can be filling the jar with are our genetic predisposition, which is these yellow circles here. These are things that are in our DNA that we have inherited from mom and dad, and that are increasing somehow this likelihood to be feeling low or sad and so on. This accounts for around like less than 40% of the factors that influence depression. The rest is about environmental factors, so stressors, things that happen to us, problems with our relationships, problems at the school, bullying, discussions with our parents, and so on. These are things that can be affecting this jar and can be increasing our chances to be feeling these depressive symptoms. But we also have these rings here on the left that are protective factors. These are things that we can also use in this jar, and I will tell you how, to be able to cope better with all these other stressors and things that are filling the jar. And it refers to things like nutrition or doing sports, having respectful relationships, good friends, and so on. So how does it all work? We are born with our jar, and we have already there some genetic factors filling that jar. With time, with experiences, we will be getting some of these stressors, these triangles, going into the jar. And sometimes we may feel that that jar is getting full. It may feel quite full at some point. And in that point, we may be actually having a depressive episode. What can we do then? Well, we can work and try to eliminate some of these stressors that were filling our jar. We can learn to manage them in a different way, to understand them in a different way, and to do things to make them either go away or work with them in a nicer way that are not they are transformed, they are not stressors anymore. Not only that, we can use these rings that I said before to put on top of our jar and make it taller, so we can cope better with the situations that we are living now and that may come again in the future. So this is the jar model. This is an illustration and a simplification of a model that is quite famous in psychology. It's called the diathesis stress model for the origins of depression. And what is diathesis? Diathesis is just a Greek word that means predisposition to something. So in this case, diathesis is just these yellow balls that I was showing before, the genetic factors. The model says that depression is a result of these diatheses or genetic factors. It's a result also of the stress, the triangles, but it's not just that. It's about the complex way in that these factors interact with each other. So it's not just putting things in the jar, it's way more complicated than that. And I was thinking, well, I will tell you about one of the studies that we ran here in our team, because we got to test this famous model that was up there from the 70s or 80s, pretty much for the first time, like directly and empirically. Why is that? Because for decades, we could only really measure things that were related with stressors, like what are the most important stressors for us to get a higher level of depressive symptoms. But genetics was pretty difficult, if not impossible, to measure until very recently. And we were lucky enough to make it possible with a study here at QIMR. So we had more than 5,000 participants that had contributed to studies in well-being and mental health. We could get a measure that told us about the genetic risk for depression that they had. We asked them about which stressful life events they had in the last year. So if they had got a breakup with the relationship or problems at work or financial problems and so on. We also asked them about the social support that they had, to what extent they felt listened and understood by friends, family, couple. We asked them also about depressive symptoms, to what extent they were feeling all of them. And we saw, and that was not a surprise, that stressful life events and genetic factors were filling that jar of the depressive symptoms, that the social support was decreasing the impact of the things that were filling that jar. But interestingly, and that is the point that I wanted to highlight with you, there was an actual interaction in between the stress and the genetic predisposition. So if you look at this plot, we have on the vertical axis the depression score, the level of depressive symptoms they had. It increased as the genetic predisposition increased also, and it also increased as stress was higher in their lives. But when taking the two things together, the impact of the on the depression score was even higher. So this is to say, it's not just the things filling the jar, is the way that in the end they moderate each other. So if you ask me, are psychological traits contagious? As a scientist, I don't think that's a right word in this context, and is not in the case of depression, because it sounds also like if you were passively getting something somehow. But as I have just explained, it's very complex. We have this jar, we can do a lot of things with that jar, and to take care about our mental health, looking at that, and not only that, but having a look to the jars of our friends and our relatives and the people we care of, to just make resources to just play with that and try to move around in a healthy way in life. Thank you.
Hello, I'm Dr. Saurabh, and I work in the Systems Neuroscience group here at QIMR Berghofer. And today I'll be talking about emotions and how we study them here in our lab. So first things first, the definition of emotions. Why is this important? Because we want to make sure that all the scientists who are studying emotions are actually studying the same thing. But unfortunately, in this field of emotion research, it's a complex field, and scientists don't precisely know what the definition of emotion is. So here I've put complex sentences and complex words about what scientists agree about emotions. But why is it so difficult to define emotions? So let's take a step back and see. And this traces back to the historical origins of the word emotion itself. So long back, the words were used in Latin and French, where it was used to mean just to move or to excite. And then it transgressed into English language, where it was used to mean bodily stirrings and bodily movements. And later on, it was associated bodily movements along with the mental feelings. But later on, in the late 18th century, it was stripped off of this bodily movements and bodily stirrings, but only the mental feelings remained. And that's why today, when we talk about emotions, it's mostly associated with the mental domains. But scientists today agree that at least there are three fundamental aspects of emotion. So one is the cognitive, which is the mental processes involved. The second is the physiological, which is the bodily changes associated with emotions. And the third is the behavioral aspect. So how do we communicate emotions verbally or non-verbally? But to study emotions, we also need to know how many emotions are there, right? So one of the ways you can think about it is a discrete categorization of emotions. So there's a fixed category and there are basic emotions, just like happy or angry or fear. And scientists have used this and found that it is common in most of the cultures around the world, where they know and recognize these emotions, and they have a name for each of this. But do you think that we only have this basic 6 emotions? Because we feel a lot of emotions which can overlap with one another. So sometimes when I'm happy, I'm also excitable. So then scientists started describing them on 2 scales, which is one is the valence and the other is the arousal. So on one extreme, you have for the valence the unpleasant emotions, and the other one is the pleasant. And for the arousal scale, it's the calm on one extreme and the excitable or arousal, high arousal on the other extreme. Okay, so we now know a fair bit about emotions, but how do we study them in the lab? So we make the participants come in the lab, and then we make them watch emotional pictures or emotional movies. And movies are really good to manipulate your emotions because that's how they are made. They can exaggerate some aspects with story and the soundtrack and the music, and they elicit really strong emotional reactions. And to detect these emotional reactions, we have lots of electrodes or wires attached to the body to measure them. For the body physiological changes, we have a setup something like this in our lab, where we attach a lot of electrodes to the arms, the head. Then there's the video cameras. Specifically, we want to measure the heart rate, which is always changing, especially during emotional movies. Then there's something called as the skin conductance, which are electrodes attached to the fingers, which measures slight variations in the sweat secretions, which also fluctuates during the movie or in your regular time. Then there's the thermal imaging camera, which measures the temperature fluctuations of the face. And then there's a regular video camera, which captures the normal facial expressions of while they're watching the movies and everything. Here's a small snippet of what it looks like in the lab, of what electrodes are attached to the body and how the signals are changing when people are watching emotional movies. And you can see there's also expressions and body changes associated with that. Okay, so this was about the bodily changes. What about the brain? How do we measure the brain changes? So for that, we use something called as an MRI scanner. You might have heard of this machine in clinics, but we also use for research purposes. So this is a very sophisticated and complicated machine, and at least there have been 10 Nobel Prizes behind the development and working of this machine. So what this machine does is it captures the images of the brain from top to bottom. So here you can see the images of the brain going from the top to the bottom. You can see some eyes here as it goes below. So this is the structural part of the brain. But we can also use this machine to know where exactly or which parts of the brain were active when a person was doing some emotional tasks. We can put the person in the scanner and make them watch emotional movies, and then we can see what parts of the brain were active. So here you can see in the red blobs what part of the brain were active when they did some emotional tasks. We can also use this machine to know the structure of the brain, so that how different parts of the brain are connected to one another. So for example, how is this back part of the brain connected to front part of the brain? And you can see the wires that are connecting these two parts of the brain in a very detailed manner. So this is essentially a Google map of the brain. So there are some busy highways, big highways, and smaller roads. So these are the different roads which connect different parts of the brain as well. So with these streams of knowledge, the bodily physiology and the brain workings, we know some parts of the brain are really important for emotional functioning, such as here. And this is what you'll hear or read regularly in magazines. But it's also not just one part of the brain which is important for emotional processing. It's the network function of different parts of the brain working together to create an emotional experience. So most often you'll hear a region of the brain called as amygdala, which is related to emotion functions. But it's not the only region. As I said, there are many regions which are as important. So what happens when these communications between the brain and the body break down? So this is what leads to sometimes the mental illnesses like depression and anxiety disorders. So for example, in depression, the bodily physiology doesn't change at all. And in anxiety disorders, it's the opposite. The body physiology is way activated. There's always high heart rate and high sweat secretion and those things. And scientists are working to exactly find out how these workings break down. And lastly, I just want to ask, are there emotions then contagious? And the answer to this would be yes and no. Some of the emotions sometimes can be contagious. For example, when you have a friend who receives a happy news, you'll get happy for him and momentarily you'll be happy. But that doesn't last days and months. It depends on your emotion and your surroundings. So the answer to this would be both yes and no. So I'd conclude by saying that emotions are really fundamental to us as creatures. It's essential for social interactions, our daily lives, happy and sad memories. And scientists are working every day to find out exactly how they work and how they break down. And I hope that some of you can also take part in your future working on this fascinating field. Thank you.
So my name is Dr. Katrina Grasby. I work here at QIMR Berghofer Medical Research Institute in the Psychiatric Genetics Lab. And I'm here to talk about intelligence and individual differences. So I work in the field of behaviour genetics, and it's a field that investigates now why are people different and what is the genetic contribution, how are we genetically different and how does that influence how we are different from each other. So individual differences in intelligence have actually been studied extensively over the past century by many different people. But what on earth is intelligence? So there are lots of definitions. People have come up with thousands of definitions from a personal point of view, but even the dictionaries have slightly different definitions. They all, however, have things in common, which is that it is our capacity to learn, our ability to reason, how we are able to plan and to make decisions, and eventually to solve problems. And when you put all of these skills together, they are being used in order to adapt to our environment. So, intelligence has been something of interest to Western philosophers for thousands of years. The Greeks were interested in it, but it came down to a French guy in the early 1900s who really put together the first intelligence test. What happened is the French government mandated that all kids had to go to school. And they employed two researchers, Binet and Simone, in order to put together a test to be able to identify which kids were going to struggle in this new school system in order to be able to provide educational support to them so that then they would be able to succeed. So this test that was developed was really focusing on cognitive abilities, like things like attention and memory, figuring out how people were going to be able to then learn acquired skills like reading and mathematics. So when we had this original test, the Binet version really gave people a mental age. So this was a score that is not what we really use today. Instead, a German psychologist by the name of Stern, he recalculated the way that we score intelligence tests, and we now have an IQ quotient, which is what you would be more familiar with. An IQ score of 100 is essentially average intelligence. So fairly soon after the French had put together this test, it got basically exported or imported to the United States, and we have a professor by the name of Turnman at Stanford University, and he translated it. He didn't just translated it, but he actually made adjustments to the test in order to make it appropriately culturally for the United States, the people living in the United States. So this is actually one of the very first examples of, or it's an early example of a cultural adaptation of a psychological test, because culturally appropriate tests are very important when it comes to considering measuring things like intelligence. So a couple of decades later, another American by the name of Wechsler, he developed the most widely used intelligence test that we have today. One for children, one for adults. So we've got further refinements happening with these tests. Now, Wechsler didn't think that a single score was an appropriate way or a very good way to measure something as complex as intelligence. Instead, he had quite a large number of cognitive abilities being tested, and these tests all clustered together into groups from which he formed subscales. So one of the subscales that we have in the adult test is called verbal comprehension. Now it measures the ability to access vocabulary, to express yourself meaningfully, and to solve verbally presented problems. So for example here, someone would be asked, what is the thing in common with these paired words? I mean, the test is a lot longer than 6 questions. This is just an example. The next subscale that we're looking at here is called perceptual reasoning. So it's going to be asking people to assess their ability to understand visual information and to solve visually abstract presented problems. So this is an example where you would need to choose which of these images below best fits this box up here. Another cognitive ability is going to be our working memory. Now this is our capacity to keep chunks of information in our mind and then perform manipulations on it to solve problems. And then also our processing speed. Now this is how fast are we able to take in information, make sense of it, and then respond to it. So all of these subscales are useful in and of themselves, but they can be combined together to give you a full score. But what makes a test a good test? So one of the characteristics that you need a good test to have is the test needs to be standardized. You need to be able to compare a person's score to a comparison group. You need to be able to place that score meaningfully into the distribution of how people perform. You also need a test to be reliable. So that means that it has consistent measurement of a particular trait, like intelligence. You need for it to be valid. That essentially means that the test is measuring what it says that it's measuring. If it says it's measuring intelligence, it's measuring intelligence. It's not measuring something else like, you know, how affable you are. Terrible example. Before I go on to that, if you apply a test outside of the context for which it was standardized and validated, it's no longer going to be terribly accurate. It's going to become biased. Your context of how a test is developed and then how you interpret it is incredibly important, particularly for psychological tests. And unfortunately, intelligence tests have a very dark history. They have been misused extensively over the last century. So one of the guys who was very first interested in actually studying intelligence, and this was in the late 1800s, his name's Galton, and he did lots of amazing things and contributed a lot to science, but he is also the guy that coined the term eugenics. Now eugenics is essentially saying, I am going to control other people's reproduction in order to influence the traits that go forward into future generations. This is starting to sound a little bit nasty. So in America, they passed into law and it went to the Supreme Court and it got passed at their Supreme Court in order to be able to sterilize people who were considered feeble-minded, which was the language of the day indicating intellectual disability. In Nazi Germany, it gets even worse perhaps. They introduced into law that you could kill children who had an intellectual disability, physical disability as well. But we're interested in intelligence in this course. Intelligence tests have also been inappropriately interpreted and used to advocate for racial segregation, to make sweeping and inaccurate statements about intelligence differences between races. Now, I mention these things because I think it's important to highlight how intelligence tests have been inappropriately used, to acknowledge that history, but to be aware that we can use them appropriately. We can interpret them for that which they were designed to do and in the context that is appropriate for them to be interpreted. My research isn't actually in the ethics of intelligence tests, My research is in the genetic influences on intelligence, but I think having that understanding is valuable moving forward because genetics is interlaced with intelligence in the way that these tests have been inappropriately used. So what I'm actually doing research in is looking at what are the genetic influences, how do we all vary in our genetics and how is that contributing to variation in our intelligence? So one approach to sort of study this is to look at a population, to look at a whole group and say, well, how much of all the differences in that group is due to all the differences in our genetics? And this is then going to give us a value called a heritability statistic. So if the variation in this group, all the variation is 80% heritable, then that means 20% of the variation is left up to other factors, like environmental factors. Now, one of the key ways that we actually look at this is one of the main methodologies is to look at twin and family studies. Now this gives us the opportunity to look at how similar family members are on a particular trait like intelligence and how similar they are genetically, and then we use complex maths to get a calculation to get a value of heritability. So heritability estimates of intelligence range from 30 to 80%. Now that's a pretty big range. So what on earth is going on? So what we find from studies that have been done across lots of different countries, across lots of different groups of people, is that heritability is changing with developmental time period. So this means the amount of variation that occurs in intelligence is being influenced to a different degree by the genetic variation of the people who are being sampled. Now their genetics isn't changing, but how their genetics is operating and the extent of how this is influencing the outcome of intelligence is what is changing. So we find that in infants, genetic variation isn't contributing an awful lot to the total amount of variation in intelligence of infants. I mean, that's pretty hard to measure anyway. But as you get older, it matters a lot more, going all the way up to about 80% in the variation of intelligence among adults. Now, what is taking up the rest of that is environmental variation. Now, this high heritability in adults compared to infants is actually very consistent with some developmental hypotheses, which say that in more nurturing and supportive environments, you're going to have your genetic potential be reached, but in inadequate environments, if people aren't getting shelter and they're not getting enough nutrition or enough food, then you're going to have a constriction, or your genetics aren't going to be doing as much as they can be doing if they had the necessary environment around them to support their expression. Now, not only is it a developmentally relevant consideration, but we also have the environment interacting with our genetic influences. So, some work that was conducted in the United States found that the heritability changed as a function of socioeconomic status. Now, this indicates that this negative value of 2 is 2 standard deviations below the average value of socioeconomic status, and this is 2 above. So we're looking at people who have low socioeconomic status down here, high socioeconomic status here, and again we have lower heritability estimates when you have an impoverished environment consistent with those developmental hypotheses, and we have higher heritability more genetic variation is accounting for, more variation in intelligence is being accounted for by genetic variation among the people who have all of the environmental resources of the high SES group. So when we were talking earlier about intelligence and the context mattering, heritability takes that up a level. Context is incredibly important. We're looking at age context. We're looking at country context here for the United States, because what they found is that this social economic status interaction with the genetic effects on intelligence isn't evident. It's not being replicated in Europe and Australia. I mean, this line here might suggest actually a little bit of an opposite effect, but we find that statistically this is not significant. There's essentially no differences across this distribution of social economic status. So context is important. So this is really interesting to me, at least, like how much heritability is there of a particular trait like intelligence, but it doesn't tell us which genetic variants matter. It tells us how much genetic variation influences variation in intelligence, but not what genes matter. To get at that, we need to go with a different method. So this is genome-wide studies. This is, or a genome-wide association study. This is where we actually go and have a look at genetic variants. So having a look at these images here, this is a depiction of DNA, and these crossbars are representing, like this is a single genetic, potentially variant. There's millions of these that differ among humans. I mean, a lot of them are going to be the same because we're all humans, but millions of these are going to differ among humans. So to take an example, take at this particular location in our DNA, some people have a red allele and some people have a yellow allele. And I've just used that colour to represent, like maybe red is representing adenine and yellow is representing cytosine, and they're just 2 of the 4 nucleotides that make up our DNA. And then what we're going to do is we're going to get a big group of people together, we're going to find out what they have at that particular location. Is it a red allele? Is it a yellow allele? And then see whether or not that one of those alleles is associated with higher intelligence. I mean, maybe neither of them are. And we do that at about 1 million or more points across the DNA. And this is what was found in a very large study. There's over 250,000 people who went into this study. This here, each of these bands, these blue and red bands, are representing the different chromosomes, 1 to 22. And then the little dots, they're the genetic variants that were tested. And the ones that scoot up, the ones that spike, all of them are associated with intelligence. So those are the points all across our DNA that are associated with intelligence. So one take-home message from this particular study as you look at that plot is to know that it is not one gene. There is no such thing as an intelligence gene. No one gene is going to cause someone to be more intelligent or less intelligent. We have a lot of genetic variants all across our DNA contributing to influencing how we are performing on intelligence tests. So thank you all for listening to me, and if you have any questions, feel free to enter, for myself or for any of the other speakers, feel free to enter them online and we'll do our best to answer them, and hopefully we will welcome you all back to the Institute sometime soon.